Now let's have a look at the data we're going to look at today. And just to change things up a little bit, we're not looking at the same data set as last time, the dependency data. Today we're going to look at gene expression data, but they come from the same source, from the DepMap data set. So here we have again data for over 1,000 cell lines, cancer cell lines. But instead of looking at how important the genes are for the proliferation, we just look at how highly expressed these genes are. So the values here, we have metadata again, like cell line names, disease type, lineage, and things like that. And then we have our 17,000 and something genes. And for each gene, and each gene is a column again, so the data are exactly structured in the same way as the gene dependency data. But here the values now mean how highly expressed the genes are. And these values are called transcripts per million. So that means how many transcript per million transcripts does that that gene is that gene counted in? And the values are normalized in a log transform. And I don't have time to explain you that in depth, but if you're interested, go to the DepMap dataset. There's a lot of explanation for this type of measurements and how they are calculated. Um, but basically, the higher so the value is not, there's no negative values here. 0 means there's no counts. That means there's no messenger RNA for this particular gene present. And the higher the value, and the value ranges between, the highest values will be about 20 or so. So between 0 and 20, if it's 20, it means it's very highly expressed. If it's close to 0, it means the expression is very low. Um, and yeah, I think that's all you need to know about this data set. We're going to have a look at it in a minute. So before we go into the data, I just want to briefly talk about the whole concept. So if you have a new data set like that, what do you do first? And this kind of work is called exploratory data analysis, or EDA. And there's two aspects to it. One is inspection and then also cleaning. I have done the cleaning already for you, so you don't have to do it again. But this is usually a big part of working with the data, is preparing them for inspection. And first we need to read the data in, and then we do the things that we did already last time. We look at the shape, we just look at the data types. But then one important thing is to check, are there any null data? We usually want to get rid of null data, and there's different methods for that. We want to get some basic statistics, and then just explore, especially if we have categorical values, we can do a quick check, like what are the categorical values, like for example, what disease types do we have? And then there is one command that will go in detail, which is group by, which is really useful to get a quick statistical analysis and separating different groups out. This will be the first part of the lecture. And then you learn the main new skill this lecture, which is visualizing patterns using matplotlib. So this is one of the major Python libraries to generate figures from data. And we're going to learn this step by step using this EDA approach. So these are typical things you do to have a look at the data, and visualizing these things will give you an access to patterns that is very difficult to see if you just look at the numbers. Yeah, so this is what we're going to do. And this is a very basic skill you should develop as a data scientist, which helps you to catch errors, to see what are outliers, whether the data are messy, whether it's worth pursuing with this data, and then to form hypotheses, to query the data quickly, and then you can go into much more in-depth analysis and start making presentation-ready figures after that. So this EDA mindset is really important to develop, that you have a quick tool to get comfortable with the data, to have a look at it, to explore what's interesting, what are the outliers. Often the outliers are the most interesting thing. What are the patterns in the data? And then form your hypothesis and formulate your question, and then you can go into much more in-depth analysis with that.